You know, when you hear the word biotechnology, your mind probably jumps to scientists in super clean labs, maybe messing with DNA. And hey, you're not wrong. But what if I told you the whole story actually started thousands of years ago with something as simple as a loaf of bread and a glass of wine? Okay, let's get into this.
So the first thing you got to understand is that biotechnology basically has two totally different phases. On one side, you've got ancient biotech. This is what our ancestors were doing for centuries, right? Picking the best animals to breed, domesticating the hardiest crops, and figuring out that yeast makes amazing bread and, of course, alcohol. Then, on the other side, you have the modern era. This is where we figured out how to work directly with DNA, the actual code of life itself. The goal has always been the same: use biology to make our lives better. But the tools, man, they couldn't be more different.
So what's the official definition? It's pretty straightforward. It's just the technique of using living things, or their products, for human benefit. But the term itself, it isn't ancient at all. Turns out, it was only coined back in 1919 by a Hungarian engineer, Karl Erecki, who's now pretty much known as the father of biotechnology.
All right, let's start by looking way back. This first part is all about how this ancient tool became a modern science. We're going to explore how humanity's been tinkering with life for our own benefit for a very, very long time.
Now, what's really wild about this timeline is the pacing of it all. For literally thousands of years, during the ancient era, all our progress came from just watching and waiting. You could call them happy accidents. Then the classical era kicks in, and you get brilliant minds like Louis Pasteur and Gregor Mendel who start figuring out the science behind it all. But look at that huge jump after 1950. That's not just progress, that's an explosion.
So what caused that explosion? Well, it really all comes down to this one single year, 1953. This was the moment, the absolute game changer. This was the year James Watson and Francis Crick laid out the double helix structure of DNA. For the first time ever, we could actually see the blueprint for life.
And that discovery, well, it kicks off our next big chapter. Unlocking the DNA code wasn't just another scientific breakthrough. It was like someone handed us the key to a library that contained the instruction manual for every single living thing on planet Earth.
And this brings us right to the heart of modern biotech, genetic engineering. What is it really? It's the ability to directly manipulate an organism's DNA. We're not just breeding the best cows together and hoping for the best anymore. Now, we can go straight into the source code to change a characteristic or even add a totally new one.
Okay, but how in the world do you edit a code that's invisibly small? Well, the huge breakthrough came when we discovered something you can think of as molecular scissors. They're actually proteins called restriction enzymes, and they are incredible. They exist naturally in bacteria as a defense system against viruses. But here's the magic part. They don't just cut DNA randomly. They look for very specific sequences of code and cut only there. Scientists realized they could use these tiny, precise scissors to cut and paste genes with unbelievable accuracy.
So how does this all work in practice? It's basically a biological cut and paste job. Step 1, you find a gene for a trait you really want, let's say pest resistance in a corn plant. Step 2, you use those molecular scissors, the restriction enzymes, to snip that gene out perfectly. Step 3, you take that little piece of DNA and insert it into a new organism. And finally, step 4, the new host organism reads those new instructions and starts expressing that new trait. It's amazing.
Armed with this powerful new toolkit, scientists could literally start rewriting the book of life. This next section is all about the incredible things that became possible once we had the ability to engineer DNA. You know, the applications are so massive and diverse that scientists actually started color-coding them just to keep everything straight. Green biotech, that's all for agriculture, making our crops better and stronger. Red is for medicine, new drugs, gene therapies, you name it. White is for industrial stuff, like using tiny microbes as little factories. And blue is for everything aquatic. The big takeaway here isn't just the variety, but the fact that biotech is becoming a foundational tool across almost every industry out there.
Let's look at a few real-world examples, because this is where it gets really cool. Back in 1994, we got the Flavor Saver tomato, the first genetically engineered food ever approved, and it was designed to stay fresh on the shelf for way longer. Then there's golden rice, which was engineered to produce vitamin A to help fight malnutrition in developing countries. And what about BT cotton? It actually produces its own natural insecticide, which massively cuts down on the need for chemical pesticides.
So, as you can see, this isn't some far-off science fiction. Let's talk about how this science is already woven into the fabric of our world, shaping our food, our health, and our environment every single day.
Here's the bottom line. Biotechnology has moved out of the lab and into our daily lives. We've gone from just observing nature to intentionally designing biological solutions for some of the biggest, toughest challenges humanity is facing.
And what kind of challenges are we talking about? Well, how about creating crops that can actually survive a drought or a heat wave? Or developing clean biofuels to replace fossil fuels? What about helping to save endangered species by preserving their genetic blueprints? And, of course, a huge one, developing life-saving vaccines. The revolutionary mRNA vaccines for COVID-19, that was a direct result of decades of biotech research coming to fruition right when we needed it most.
All of this brings us to our final section, the next chapter. With all this incredible power, where do we go from here, and what kind of deep questions does it force us to ask?
So we've gone from ancient brewers to modern gene editors. We've seen how a very basic human desire to make things better has evolved into a science with almost unbelievable power. The ability to edit life itself is now in our hands. So the ultimate question really isn't what can we do anymore, it's what should we do? With this power, what future are we going to choose to build?